Every single book which have toppers suggested, but nothing worked for me.
I was like कि नहीं होगा छोड़ देते हैं, नहीं हो रहा है तो क्या फ़ायदा इनको दोबारा पढ़ने से.
I would like to thank StudyIQ. I got connected to StudyIQ.
StudyIQ ने मेरा मेंस तक मैंने साथ दिया, उसके बाद भी उन्होंने मेरा साथ नहीं छोड़ा.
I have taken this mock at StudyIQ.
And I would recommend StudyIQ.
Yes, I will definitely recommend StudyIQ.
Hello everyone, I am Ankit and you're watching StudyIQ IAS English channel. In our today's video, we are going to understand the facts about DNA identification. And why are we discussing DNA identification? Because last week, as you know it already, Air India plane which was traveling from Ahmedabad to London, it crashed few minutes after it took off. And the consequence of this air crash was that the people who were traveling in the aircraft, now their remains are burned beyond recognition. So the authorities, they are relying on DNA analysis, that is deoxyribonucleic acid, and its analysis to identify the victims and hand over the remains to their respective family members. But have you wondered how this DNA analysis actually works? Well, today I'm going to explain everything to you in great detail. First, let us understand the context. Now, after Air India plane, which was a Boeing 787 Dreamliner, it crashed in Ahmedabad, the authorities, they are now using DNA analysis to identify the remains of those who have been killed in the accident. Because you understand, those bodies have been burnt beyond recognition, so you can't identify them solely by looking at them. So what you need to do is conduct DNA analysis so as to identify which body belongs to which people and ultimately hand them over to their relatives. Now, the DNA samples from the victim family members have already been collected by the authorities, and few remains have already been identified using this DNA analysis itself, and it also includes the body of ex-CM of Gujarat, that was Mr. Vijay Rupani, who is also one of the victims of this plane crash. Now, the DNA identification, when it comes to identification of bodies, it is a gold standard, especially after a mass fatality accident where it is not easy to identify the bodies. So, previously also in many of the related fire incidences, the authorities have relied on DNA identification because this is a best way to identify the body of a human, right? Now, first, let us understand the process that it is taken to identify the DNA. First, obviously, you can understand you need to collect and store the samples, and as soon as the individual dies, their DNA begins to degrade. And over the time, the degradation can make it difficult. And if the DNA is degraded so much, sometimes it can also be impossible for the DNA to be analyzed. And the extent of degradation of DNA depends on two factors. First, what kind of a tissue are you taking DNA from? Whether it is being taken from a soft tissue like muscles or skin, or has it been taken from a hard sample or a hard tissue like your bones and teeth. Similarly, the conditions also on which the body is kept plays a very important role in determining the degradation process of the DNA. Whether has it been stored in a hot and humid condition, or has it been stored in a cold or dry condition. So those two factors actually determine the degradation process. So let us understand these two processes in great detail so that we know the entire process of collection and storing of samples. As you can understand, the DNA sample, it survives much in a better way in the cold and dry conditions as compared to hot and humid conditions. And to preserve these DNA samples, these needs to be collected as soon as possible, and once collected, they need to be stored in a cold and dry environment. So for that, the samples are stored in a frozen format in -20 degrees centigrade, and in case of soft tissues like skins or muscles, etc., you need to store them in a solution that consists of 95% of ethanol, right? So this is the process to store a sample in a cold and dry environment so that the degradation of DNA is prevented, or at least is slowed down, right? Another factor, as I told you, it determines what kind of a sample have you taken. So the DNA which is taken from soft tissues like skin and muscles, it degrades much faster as compared to samples taken from hard tissues like bones and teeth, right? This is because cells which are there in your hard tissue, these are largely protected from degradation due to putrefaction and also prevents from decomposition, and that is why the forensic investigators, they usually collect the DNA samples from hard tissues itself, and this is to prevent the rapid degradation of DNA from happening. So this was the first process of collecting and eventually storing these samples. Now after you are done collecting and storing these samples, the next process is the analysis of this sample so that you are able to ascertain the DNA and attribute it to an individual, right? So for that, authorities depend upon many processes which we'll discuss, need not worry. Now let us discuss few instances of where collection and storing of samples have been quite difficult. Now you understand the collection and storage of samples may also depend on the scale of tragedy. So if the scale of tragedy or any incidents that has happened, it spreads over a large duration or for a large geographical extent, it makes it difficult for the investigators to collect the samples. For example, during the plane crashes that happened during the 9/11 terrorist attack in US, it took authorities as much as 10 months to collect the entire DNA samples from nearly 3,000 victims. That is why after a plane crash, it can take few days to months before you are able to collect all the DNA of the victims that have been perished in those incidences. And to identify the collected DNA, who it belongs to, you need to also reference these samples from the samples of their biological relatives, right? So for that, the best course of way is to take the samples from the relatives that are parents and children of the victim, because they share a majority portion of the DNA from the victims, and that is why you'll be easily be able to attribute and reference those samples from their victims, right? So this is the process of collecting and storage of samples. Now let us understand what are the different methods to analyze the DNA, right? First, and the best way to analyze the DNA is known as short tandem repeat analysis or STR analysis. Now in this method, what the investigators do, that they ascertain short repeating sequences on their DNA. And what kind of DNA is utilized in this particular analysis? There you take DNA from the nucleus of a cell of a particular human being, and you analyze nuclear DNA in this process. So after you analyze 15 or more of these hypervariable regions of DNA, and if you are able to understand the short repeating sequences in a particular human being, you'll be easily be able to ascertain the family relationship with relatively high degree of confidence. So that is why this is the best way to analyze the DNA, but there is also a challenge associated in it. So that once you take the sample to use this kind of a DNA analysis, you need a sample from nucleus of a cell. That is, you need to ascertain the nuclear DNA. So if you are not able to take the DNA or a nuclear DNA from the human being, then you will not be able to conduct this particular kind of test. But in cases of degraded sample, there are other methods also which we'll understand. So in this regard, the second method is known as mitochondrial DNA analysis or mtDNA analysis. Now in this method, this people or the investigators, they take this method or use this method in cases of nuclear DNA gets degraded or it is unavailable for certain reasons, right? So what scientists do is that they obtain the DNA from your mitochondria, and as you can understand, mitochondria is more commonly referred to as powerhouse of the cell, right? So you obtain mitochondrial DNA from these mitochondria, and then you ascertain and identify the samples and cross-reference with your relatives. Now there is relatively it is easier to conduct this kind of an analysis as compared to STR analysis. Why? Because mtDNA is present in multiple copies within a cell, so it is easier for the investigators to recover this from human remains that might even not be some well preserved in this. Now you understand where you get a mitochondria from. Mitochondria in a human being is inherited from your mother. So for identification purposes, the investigators need the reference samples from your mother side of relatives. It can include your mother or the relatives from your mother side. That may be their brother, their sisters, your grandmother from the maternal side. So that is why you need samples from the maternal side of the family, so that you are able to establish the maternal line of inheritance in this case, right? So this is one of the challenges in this regard. It is easy to collect mitochondrial DNA, but when it comes to cross-referencing, you need the DNA samples from your mother side of the family, including your mother, right? So this was the second instance of conducting DNA analysis, that is mitochondrial DNA analysis. Now the third analysis method is known as Y chromosome analysis, because you understand humans, they are made of two type of chromosomes, that is X and Y. So a biological male, they'll be having a XY chromosome, whereas a biological female, they'll be having XX chromosomes, right? So in this method, a panel of STR on Y chromosomes is ascertained, which is passed to a particular victim, most likely by their fathers, because it is only the male side of your family that is carrying the Y chromosomes in it, right? Not your mother side of the family. So that is why this sample is examined to match the remains with your male side of your relatives, not your female side of the relatives. And this method is useful when close relatives are not available. So you can take sample from any of the relatives belonging to your father side or your paternal line, right? So that is why you can take samples from your father, your paternal uncle, your paternal male cousins, and this will be useful in matching your DNA in Y chromosomes analysis. Understood why? Because only males carry the Y chromosomes, because females, they don't. Understood? Now, the fourth way to ascertain your DNA is known as single nucleotide polymorphism, SNP analysis. Now, this is typically used when the DNA sample is highly degraded, and in this analysis, your SNP, that is single nucleotide polymorphism DNA sequence, is analyzed. Now you understand, in your human being DNA, there are 4 nucleotide bases: adenine, guanine, cytosine and thymine. And the way these are arranged in a human being, they differ individually. So the way DNA sequences are arranged in my DNA, it will be different from yours. Similarly, it will be different for other people also. So these SNA sequence, these are unique to each person and they can be used for identification purposes. And the sample that you need in this case can be taken from victims' personal belongings like the toothbrush or even their hairbrush. So the samples are retrieved from your personal belongings and then your sample is matched using the SNP method. And in this case, it is not maybe as effective as the STR analysis, but the thing or the best aspect about this analysis is that you can take samples directly from the human being or their belongings, right? So these are the 4 methods in total that help you analyze the DNA, and these depend on the kind of sample that you've obtained. If you have taken a good sample or a nuclear DNA sample, you can easily opt for a best method, that is the SNP analysis. But in case you don't have a good quality sample, you can opt for other methods that I've told you, that is mitochondrial analysis, Y chromosome analysis or the SNP analysis, based on the kind of samples that you take. And for collecting the samples, you need to be as quick as possible, because as soon as a human die, their DNA samples, they start to degrade, right? So these are the things that you need to know before ending our today's session. Now, there are questions that I've attached to our today's analysis, but before attending those questions, there is something that I need to tell you. First, that how is DNA different from RNA? What are the various aspects of DNA? We cover them in great detail in our Pratigya morning batches, right? Now, up till yesterday, we were taking admissions for evening batch, but now those admissions or those batches have been full to brim. So we have now started to take admissions for morning batches as well under the P2I Foundation Pratigya batches, and this batch is expected to start on 23rd of June. To take admissions in these batch, what you need to do, go to our website, apply the code ATS Live. Why? Because it will get you admissions at this amount. How to apply the code? What are the various features of the course? Let me tell it to you, right? Now for that, what you need to do, you need to go to our StudyIQ website, right? Once you go to our StudyIQ website, you will be needing to create an account here by simply logging in with your mobile number. Once you're done logging in with your mobile number, I understand many of you who are watching the video might not be comfortable with Hindi as your teaching language, right? So in that case, if you're not comfortable with Hindi or with bilingual batches, what you need to do, select all language and opt for English. In the English batches, we are offering 2 kinds of batches. First, that is the morning batch of 2026. It is for the people who are going to attempt your prelims examination next year in 2026. But if you plan for your exams later, that is in 27 or 28, you can opt for long-term evening batches. The features of both these batches are same. The only difference is the duration of these batches, right? So to take admissions in the 2026 batch, what you need to do, click on the leftmost tile here by clicking buy now. As soon as you click buy now, you'll see that this course is available at 37,999. But I'll tell you a way where you can get admissions to these batches at a discounted rate. What you need to do, click on buy now. How to get discounts, I will tell you, right? Right here, there is an option to add promo code. Click here and on the left, write ATS Live. This is the same code which is appearing on the bottom half of your screen, that you can opt for any course that is there in StudyIQ batch, right? So once you're done writing the words ATS Live, click on apply. As soon as you click on apply, you'll realize that the discount of 7,000 rupees is now applied and you get diamond batch at 30,999. Platinum is at 27,500 and gold is at 19,999. Obviously, you can take admissions in either of these 3 batches, but if you ask my opinion, I'll recommend you Diamond Batch. Why am I recommending Diamond Batch? Because Diamond Batch comes up with many features that are not available in either Platinum or Gold. For example, you'll be getting a mains residential program. So if you clear your prelims examination next year, you'll be brought to Delhi and every of your expenses, food related, lodging related, study related expenses will be borne by StudyIQ. And you'll be kept in an environment that promotes competition, that promotes excellence on your part, right? Also, you'll be getting a prelims residential program, which is same as mains residential program, only before prelims examination. Also, you'll be getting a selection box, which is all the books which is necessary from your exams perspective will provide you that. And you'll also be getting direct guidance from faculties under Margdarshan. Similarly, if you clear prelims, all of your course fees, yes, all of your course fees will be refunded back to you because you're getting scholarship in the Diamond Batch, right? And scholarship for Diamond Batch also includes a 11,000 rupees financial reward in case you top your monthly test, right? And all of these benefits that I've told you, mains residential program, selection box, prelims residential program, scholarship is available exclusively in Diamond Batches, right? So that is why I was recommending you Diamond Batch. You can take admissions to Diamond Batch at this rate, but please do not forget to apply the code ATS Live because only then you'll be saving 7,000 rupees of your money. If you still have doubt what are the various features of StudyIQ courses that are there in Gold, Platinum and Diamond, and if you want to compare and contrast them, this is the slide that you need to take a look onto. I will upload them in the notes. You will realize it, okay? Now coming back to the question, this is the first question that I've curated on our today's discussion. This is modeled exactly on a DNA based question that appeared in 2023 prelims. And I have another question for you before this session ends is this one. Single nucleotide polymorphism analysis is typically used in which of the following cases? For both of the answers, please let me know the correct answer in the comment section. I will tell you the right answer in case you answer it correctly by simply hitting the like button so that you know your answer is correct. And if you want to download the notes, these will be available on my Telegram channel that is called as ATS Live. So go to Telegram on the top right hand corner of our app. You can enter the word ATS Live, you'll find the channel. And if for some reason you still aren't able to find the channel now, this is the QR code that you need to scan. Subscribe to the channel if you still haven't and download the notes. So this is all from my side for today. We'll see each other again tomorrow. Please have a very good night.
Every single book which have toppers suggested, but nothing worked for me.
I was like कि नहीं होगा छोड़ देते हैं, नहीं हो रहा है तो क्या फ़ायदा इनको दोबारा पढ़ने से.
I would like to thank StudyIQ. I got connected to StudyIQ.
StudyIQ ने मेरा मेंस तक मैंने साथ दिया, उसके बाद भी उन्होंने मेरा साथ नहीं छोड़ा.
And I have taken this mock at StudyIQ.
And I would recommend StudyIQ.
Yes, I will recommend, definitely recommend StudyIQ.